All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. This is an impromptu discussion that I wanted to do out of turn. Today is a Saturday, but here is the discussion. There are two studies that are very important at this time. I was watching news and there was a lot of panic that BA.4 and 5 are spreading fast and they're causing lots of cases and so on. And I think it is fair to see that it is going to happen to all of us, that we are going to be infected by BA.4 and 5. There are two studies that I wanted to share in this context. One study is from South Africa, and the other study, I believe, is from Japan. The summary of these two studies is the following, and then I'll go and I'll read through the studies. So it's summary first. The South African study, it's about 10 days old that it got published. It is about Omicron BA.4 and 5. And here is what that study says, and it is such an important thing. The study says that neutralizing antibodies produced by the infection by BA.4 or 5, BA.4 or 5, is cross-protective to almost all the previous variants of concern, with varying degree of protection. That also means that if it is so, the neutralizing antibodies are so diverse because of BA.4 and 5, that these can cover the previous variants of concern, then it is possible that these can cover the future variants of concerns. One. Second, these are my inferences. Second inference is that those of us who are worried that Delta is coming back, which I do not believe it is coming back, but if there are people who are worried that Delta would reemerge, there are mutations going back towards Delta, then you would see that this BA.4 and 5 infection is resulting in neutralizing antibodies that have very good protection against Delta. So this is a hugely important concept. The studies, for example, the Harvard study that we saw, that said that current vaccines or previous infections are not able to protect a lot from BA.4 or 5, regardless of the severity. They are protective for severity, but they're not protective for a lot of neutralization. So cases and infections are occurring. That study is looking forward study. Vaccines were made for the design of Wuhan variant. And we are now 5 dominant species after, not species, variants after. So those previous neutralizing antibodies not working on the current ones is actually understandable. Virus is mutating. Here, this study is different. It is looking at current infections antibodies and looking backwards. So that is a beautiful study. The second study and summary. This is from Japan. They found, so very interesting conjecture. Their conjecture was that the SARS-CoV-2 infection and severity is more common and prevalent in folks in higher age, advanced age. They said, we wanted to understand, why do they have a higher load of the virus? Why do they have more virus replicating? And is that something that is not happening in younger, healthy individuals? So they took the saliva from healthy individuals, just the saliva, nothing else, no medicine, no herb, no tea, nothing, just the healthy individual's saliva. And they put that with SARS-CoV-2 and the cells with the ACE2. And they found out 4 important proteins that are in all of our saliva were able to block S1 from binding to S2, ACE2, which meant people that have less dry mouth, younger people have actually, I mean, as we age, we start developing dry mouth, dry skin, and so on. Our glands that release secretions, this reduce functioning just like other parts of the body. So younger individuals, healthier individuals with good amounts of saliva were able to protect by reducing the viral load, by blocking the S1 protein from connecting with ACE2. And so this is the second study. I'm now going to go in the detail that what are the 4 proteins they found and what are the, in the first study, what is the neutralization they saw. But if you just wanted to hear what is the summary of these two, let me just give you a summary once more. One study from South Africa that shows that BA.4 and 5, when they infect someone, vaccinated or not, doesn't matter. When they infect someone, they produce neutralizing antibodies that are cross-reactive to the previous variants. My conjecture with that is that that would mean they would also be protective for future. You may be looking at a big rise in the cases, but we may actually be also looking at the endemicity with reduction in damage. For once, we all are going to be infected, and we still are at risk. However, somebody who has become infected and lived to tell that, they actually now have a beautiful, better protection compared to BA.1, for example. So now let's look at the data. This is it. And the second study is saliva itself. Now, what I'm going to do is, number one, I'm going to go over the study. Number two, I'm going to talk about the 4 proteins in saliva. And number 3, I'm going to talk about what are various ways we can increase our saliva. So let's look at these. Lilchi says, I have spit in my mouth as well. Yeah, so the ways to increase the spit. And if you're a youngster, we are comfortable with your spit. Okay, let's start. So this is drbeen.com. There is a link in the description that is for drbeen.com premium lectures, and the price would change on Monday because the current price, which is a one-time fee, my marketing team says that's not a price that gives a good perception. So we have it going today and tomorrow, and then Monday we'll change it. This is the first study. It's a preprint, SARS-CoV-2 BA.4 infection. And down in the study, they say BA.4 and 5. BA.4 infection triggers more cross-reactive neutralizing antibodies than BA.1. So because I've given you the basic gist of the study, let me go to the graph, the chart that shows this, and then we can look at some of the notes here as well. So first, I was doing this homework over this Saturday. So I actually have to give this credit. So there is a secret admirer doctor who I am a fan of, and he has been helping me when I was infected. His name is Dr. Tommy Zaharakis. I have actually requested him to come join us. He teaches, and he's a practicing doctor here in California. He sends me these studies. These 2 studies are actually sent by him about an hour ago. Then I went and I read the studies, and then I sent the message back to him, and I said, I'm going to share this right now because it would help in 2 ways. This study that I'm doing right now, South Africa, that would help us put the panic in perspective, that how much should we be panicking. And the second one, the spit one, the saliva one, would help us figure out how can we improve our chances of having a lower viral load. Okay, so look at this picture. In this picture, let's look at the left side first. That is the unvaccinated individuals. And how is this data present here? If you see here, this is BA.4 line. Above the dotted line is their test's sensitivity that it is reading. Below the dotted line, it cannot read those, the titers. These are neutralizing antibody titers. BA.4, this line here, is the optimal activity of these antibody titers. Now, when the BA.4 or 5 infection occurs, these are actually antibody titers from the patients. And then they took those plasma and they cross-reacted them to the previous variants. The researchers are awesome people. So here, this is the line for the BA.4 and 5. This line here is for BA.2. And if you see, it is a very similar and very close line. That means BA.4 and 5 infection-produced neutralizing antibodies are able to equally control BA.2. Then this is BA.1, slightly less, but still able to control it. Then this is Delta, still slightly less, but you can see the range here. The sample size is small as well, but able to control Delta too. And then here, Beta, and then here, original Wuhan variant, all variants. And this is my conjecture now that if there is the mutation, current BA.4 and 5 mutation is such that they are able to trigger the antibodies, which are neutralizing antibodies that are able to neutralize the previous variants, all of them of our concern, then it is possible, it is highly likely that future variants will also be covered by it. So this may be that we are getting infected and reinfected, and that's just our body and the virus is playing a match to finally become okay. In that process, I realize there are some studies that say that every reinfection is going to make us worse. That is the Veterans Affairs Hospital study. There are studies I have shown which show reinfection duration is long, reinfection severity is less, and so on. So there are all kinds of studies. Here is an important study for looking into the future. And at least I think it reduced my panic. So maybe it can help some of us here as well. On the right side is the vaccinated individuals who are infected. So these are breakthrough infections in vaccinated individuals with BA.4 and 5, the infection with BA.4 and 5. Of course, the vaccination is still Wuhan variant design vaccine. So here, if you see, this is the line of BA.4 and then BA.2 coverage. And if you see, because of the vaccine, there is some additional boost as well. BA.2 coverage, BA.1 coverage, Delta, Beta, Wuhan. So according to the researchers, it did not matter that you were vaccinated or unvaccinated. If you got infected by BA.4 and 5, if you were able to continue on without getting, BA.4 and 5 are still causing severe disease as well. It's just that the incidence is low, but they are still causing it. Then this is the outcome. And if I then go to the second diagram here, this was also very interesting. On the right side, this is BA.4. And what they did was they took BA.4 neutralizing antibodies and they had them attack the virions, lentivirus they used, pseudovirions of previous variants. And if you look at it, BA.4 was able to cover all of them, although they say that any number below 1. So this is the reduction, how many fold reduction was present in efficacy, you can say, or in the protection or neutralization. So below 1, they said you could consider below 1 means that the blocking of the variant is almost not there. So here, if you see BA.4, this is BA.4, I believe, then BA.1, then Delta. It has the lesser number for Delta. And then this is Beta, and this is Wuhan. So it covers everyone, except slightly lower with Delta. But still, if you compare this cluster with all the rest of them, the only variant that comes up somewhat near this is Beta. BA.1 was selfish. It did not allow neutralizing antibodies to be produced that could cover other variants. Delta was very selfish. It did not allow, the mutations were such that the epitopes were such that the neutralizing antibodies were not sufficiently cross-reactive. And if you can see it, it was not protective for others. And here is the Wuhan. Of course, if Wuhan was protective for others, then there will not be a next variant. So it was not. And here, BA.4 and 5, they kind of rise up and they're becoming a wall for at least the previous variants. Hopefully, they would become a wall for the future variants too. So these are the two graphs. Then if very quickly I can go over some of the notes here. Differentially trigger neutralizing antibodies, the various variants of concerns have neutralizing antibodies that are different produced by each one of them. Then they say BA.1, which triggered neutralizing antibodies with limited cross-reactivity. BA.1 was bad. The only two good ones are Beta and BA.4 and 5. BA.4 and 5 being 1. BA.4-5 infection triggers highly cross-reactive neutralizing antibodies. Cross-reactivity was observed both in the absence of or prior vaccination and also in breakthrough infections following vaccination. So this suggests that the next generation, so their conclusion was that if you are going to redesign a vaccine, redesign it based on BA.4 and 5. It is a better vaccine. So BA.4 and 5, I would say, is a better infection than other ones. And that I'm not proposing that we go and get the infection. These infections are still putting people in the hospital. Some of them are dying as well. So that's not what I'm saying. But BA.4 and 5, if somebody got infected, they have a better opportunity to protect themselves from the previous and the future variants. Then here is what they did. The BA.4 and 5 sublineages, they start from here. BA.1 has 30 mutations on the spike region, which reduces the neutralization capability for other variants. BA.4 and 5 sublineages, which share the same dominant spike mutations, but differ from one another in non-structural proteins, as well as the nucleocapsid and matrix genes. So their spike, BA.4 and 5, the spikes are same. The other parts of the virus are different. And they cause a fifth wave. Within spike, BA.4 and 5 are genetically similar to BA.2, but contain the 6970 deletion, additional substitution in the receptor binding domain, namely L452R and F486V, and the R493 revision. This reversion is what is causing people to say, hey, it is becoming Delta again. Therefore, compared to BA.1 and BA.2, BA.4 has shown increased neutralizing resistance to vaccine and convalescent sera and monoclonal antibodies. So BA.4-5 are resistant. We are seeing that. The Harvard study came out and said the whole world is stopped now. We and others have shown that each SARS-CoV-2 variant triggers different profiles of neutralizing antibodies. Fine, understood. For example, the beta variant triggered humoral responses and increased cross-reactivity, and consequently became a focus for the development of the vaccines. In contrast, BA.1 triggered more strain-specific neutralizing antibodies with limited cross-reactivity in individuals with prior infection. So it didn't really help others. It was just very selfish, made antibodies for its own self. Here we assess the cross-reactivity, and then they talk about how did they do that, and I have shown you the results. So this is one study. I really liked it. And thanks to Dr. Zaharakis. Now, this is the second one. And once again, I'm going to keep the high level of this one. I'll just explain one beautiful little concept in here. So this study, an Osaka Metropolitan University research group led by Associate Professor Misaku Matsubara of the Department of Veterinary Sciences, Graduate School of Veterinary Sciences. And I want to give credit to the writer here, reviewed by Emily, and I saw the author, so sorry. Anyways, here is what they found. The research group has shown that saliva from SARS-CoV-2 uninfected healthy individuals interferes with S1 and ACE2 binding in a concentration-dependent manner. More saliva, more difficulty in binding. They identified 4 proteins in saliva that bind to ACE2, and among these proteins, neutrophil elastase and histone H2A markedly inhibit the binding of S1 and ACE2. So let me explain these 2 proteins. I'm going to draw it. My drawing. Oh, I did not show this one. So this is the BA.4/5 protein. This is the antibody. And these are other variants crying and upset because it is now attacking them all as well. So with this, let's now look at the spit part of the discussion. So here is what they saw. Here is a neutrophil. Neutrophil is part of an innate arm cell. Innate arm cell mean that this cell is present in many of the surfaces of the body where the defense is needed. So let's say this is a neutrophil. And let's say it's a happy neutrophil. It's just continuously helping us and we are happy with it too. Neutro. Now, neutrophils do a lot of functions. They do phagocytosis and they do destruction of the pathogens. They do destruction of and debris clearing of the other cell debris and so on. But they have another very interesting thing. Once they are becoming active, they can release nets. These are really protein structures that are released from them. And interestingly, when these nets are released, what happens is on the nets, this is like neutrophils are fishing. So as soon as they think that, hey, we are under attack, there are some pathogens around, there is some debris around, they release these nets, neutrophil nets. And Skyfrog is here. Skyfrog, love you. Thank you for being here. I hope you're doing well. So here are the neutrophil nets. And the nets have various little enzymes attached on them and pieces of DNA, like little bait for the fish being the viruses and bacteria. So they're tiny pieces of DNA and RNA and other proteins and enzymes are attached to these nets. And the neutrophils would then release those nets in the environment, and these nets would help neutrophil catch the pathogens or debris. H2A is a histone protein. What does that mean? I had done this discussion a few days ago that inside of our nucleus, the DNA, that long string of our life, the recipe of life, that is so long a string that if you just leave it in there without properly organizing it, it will all become tangled up, just like a string of yarn. If you have a yarn, those little balls of yarn, if you take all the yarn out and just put it there, it's going to jumble up. That is the same situation for us as well. You have long DNA, it's going to jumble up. So what we have is inside the nucleus, we have proteins called histones that come in 4s, and around, imagine that these are the cotton balls. Around them, the yarn or the DNA is wrapped. This is how we keep the DNA organized and prevent it from becoming tangled. So now the question will be that when we want to make an RNA, when we want to express a gene, what happens? So there are enzymes that come, they kind of take it off, they loosen it up over there, then they open it. We make a copy of the gene that becomes messenger RNA. That messenger RNA comes out. Meanwhile, the nucleus is then, the DNA is once again tightened back up on that little ball. This messenger RNA then goes out and does its function, and we make a protein. So that's a recipe. These histones are proteins from nucleus. So the neutrophils are able to pick up those proteins from within the nucleus, because these proteins are made in cytoplasm, then sent in the nucleus. They are also present on the nets. There are pieces of debris or little tiny DNAs are also present in the net too. And then, of course, there are elastases. Elastase mean something that breaks elastic things. There are proteins that are elastic. So really, ase at the end means a protein that can break another protein. What kind of protein will it break? That is the first name. Elastase would break an elastic thing. For example, fibrinogen and collagen and fibrin threads. DNases will break DNA. RNases will break RNA. Proteases will break proteins, and so on. So elastases, pieces of DNA and H2A. Out of them, the elastase and H2A were the two dominant proteins, which when present reduce the S1 binding with ACE2 in a dose-dependent manner. So dry mouth can be a problem for viral load. And there are many ways to see if you have dry mouth or not. For example, some of us actually know that we have a dry mouth. Some of us may have bad breath, which one of the reason for bad breath may be dry mouth. And so there are ways to know if you have dry mouth or not. Now, the question is, if you have dry mouth, what can we do? So if we can do this, I had done mouth washes in the past. There is a video that I'd done, mouth washes. Today, I'm just going to very quickly go over some of the ways with helping with the dry mouth. Dry mouth is also called xerostomia or xerostomia. And there are many reasons here. I'm not going to go over the reasons. If somebody has it, they have it. Then here is how to treat this. So manage any underlying conditions that cause dry mouth. The conditions could be, number one, normal age, and number two, which aging we cannot yet treat, but autoimmune diseases can cause it, the dry mouth. Tooth decay can cause it. And then increased saliva flow, meaning we are taking the saliva away from mouth more than we are producing. There are many treatments available. A simplest treatment is drinking more water. More water will cause more secretions, which will produce more wet mouth or saliva. At the same time, drinking water also helps with the bronchial system, because the secretions inside the windpipes are important, because there are cilia within the windpipes that move those secretions upward, and in those secretions are trapped various pathogens and dust and dirt and allergens, and they are brought up, and when they reach our throat, we either spit them out or we swallow them. So that is called mucociliary escalator. That escalator also benefits from when we drink more water, because this becomes more wet, and then it becomes easier to clear the windpipes or the airways. So drinking more water not only helps the dry mouth, it would help the airways as well. So look at this. Avoid decongestants and antihistamines when possible. Again, when possible. Sip water before swallowing capsules or tablets. Take medications in the morning. So this is if somebody has dry mouth and they need to take medicines as well. What other things can be done? Ice cubes or sugar-free ice pops, sugar-free hard candy, or sugarless gum. And Dr. Zaharaki has actually sent me one more study, which I didn't get a chance to read it, so I'm not presenting. In that study, what researchers did was they mixed herbal antivirals in chewing gum, and they gave the chewing gum to the patient. And not only now the patient had their mouth coated with the antiviral, but they were producing more saliva as well. And then because of chewing gum process or activity, those herbal antivirals were coated on teeth as well. So protection against SARS-CoV-2 became better. And they said this chewing gum would have been sufficient in that study. And I have to read that study and I have to share it. I am giving you my first glance look, so it may be wrong as well. They said that chewing gum may have been sufficient to protect people from developing COVID. So artificial saliva products, toothpaste that are made for dry mouth, lip balm, cool mist humidifiers. Then avoid acidic, spicy, salty, dry, and sugary foods and beverages. Sugar, what happens is that when the glucose is pulled into the blood, it takes water with it. So it can actually cause, then if the glucose is eliminated, it takes water with that too. Alcohol, caffeine, and carbohydrates. You know that alcohol or caffeine or coffee, these are diuretics as well. So they would reduce the fluid volume, which would cause dry mouth. Mouthwashes with alcohol or peroxide, which may dry your mouth even more. Smoking. Then drink plenty of water, use a humidifier, breathe through your nose, avoid over-the-counter medications that can cause dry mouth, such as antihistamines, decongestants. Ask your health provider for that. Then caffeine, tobacco, and alcohol. The most important for me is to drink more water. And if you can drink nutritious water, meaning something with the, let's say, vegetable soups, chicken soup, fish, whatever soups of various kinds would also help, because they also have the nutrients that are needed by the cells, and they have the fluids in them to keep us from drying the mouth. There are some diseases where you can't help it. There are autoimmune diseases, for example, Sjogren disease, where there is dry mouth, dry eyes, and dry vaginal surfaces. And we can't really do much about it other than the fluids or the supplements from outside. So these are the two studies. Thank you very much for being here. I was excited about them, so I interrupted your weekend. My apologies for it. I hope that first study gives you hope, and the second study gives you an idea of how, if you're going to be exposed at some point, if you are not infected yet, regardless of your vaccination status. If we see our president was double primary series, double boosted, and is infected. So there is no guarantee with the vaccine that infection will not occur. Important thing is, how do we protect ourselves? So thank you very much. Please like, subscribe, and share. There are links in the description if you would like to support this work. There is a link you can buy Drbeen account. The prices will change on Monday. That's one. You can become a part of Locals or Patreons or Substack, or you can use PayPal, or you can buy me a coffee. With this, thank you very much. And I would see you on Monday, unless there is another study. So talk to you later. And thank you very much for those who joined me this morning on Zoom walk. Every Saturday, 7 a.m. Pacific time, we do a walk together on Zoom. Bye-bye for now.